growth and reproduction. Ontogeny of a pseudocymbaline trilobite. This study involved scientists conducting observations on a large sample size of fossils in order to better understand the ontogeny of late mid-Fenian pseudocymbaline trilobites, a group of marine invertebrates. By examining and comparing multiple remains of a species of trilobites, they are able to piece together the ontogeny of this particular protid and the changes and adaptations it underwent. The question being tested is, will the recovery of O. prima's earliest larval stages, in addition to later growth stages, allow for the first complete growth series of a pseudocymbaline trilobite to be constructed? The procedures involved in the study required the observation of changes in physical growth throughout the ontogeny of protids. All observations and measurements were taken and recorded via a Nikon mesuroscope. 10% formic acid and etching tools were used to remove the specimens. This species was found in southeastern Morocco, and the age of the sample ranges from larva to the later growth stages. The first stage of ontogeny is the pre-metamorphic stage. Over 50 remains are recovered. Only one pre-metamorphic stage is recognized and observed. Some of the notable characteristics include short triangular axis, devoid of transverse furrows, and slightly rounded posteromediales. The second stage of ontogeny is the post-metamorphic stage. Approximately 60 remains were discovered. Mostly broken specimens were recovered, however 8 were well preserved. Only one metaprotaspid stage is observed as well. Notable physical characteristics include slightly elongated, posterior border furrow is widened, and occipital furrow deeper and wider. The third and fourth stages of ontogeny are the meraspid and holaspid periods. The meraspid stage occurs directly after post-metamorphosis, then transitions over time to the holaspid period. Notable comparisons of the meraspid to holaspid include the genal field enlarging and expanding backwardly posterior to the eye, also initially bearing 2, then 3 and 4 terrace ridges. Noticeable comparisons to the metamorphosis stage is the glabella elongates, the occipital furrow is deeper, and the occipital ring is broader. Below are some of the general trends seen throughout the ontogeny of O. prima. Some of these include post-axial field slightly decreasing in width, 8 to 9 apparent ribs in youngest meraspids, and the posteromedial notch progressively disappears. Below is a scatter plot demonstrating and comparing the various lengths and widths of multiple species of trilobites. In conclusion, the general trend during the ontogeny of O. prima is the flattening of the labrygina, allowing the enlargement of the labryginal field and the shift in the orientation of the flattening genal spine. Basically, the ontogenetic changes result in the flattening of the exoskeleton. The observations were conclusive with the hypothesis that the recovery of O. prima's earliest larval stages, in addition to later growth stages, allowed for a complete growth series of pseudocymbaline trilobites to be constructed of its ontogeny. This study also provides significant information on the evolution over time of the early ontogeny of protid trilobites.
Second paper is on the sexual maturity in growing dinosaurs and how it does not fit reptilian growth models. The 3 dinosaurs that are being tested are the Tyrannosaurs, the Allosaurus, and the Tyrannosaurus. Some background information. The medullary bone, which is usually found in birds, was recently discovered in 3 species of dinosaurs. The 3 species that were taken part of the test were the Tyrannosaurus, the Tyrannosaurus, and the Allosaurus. Tyrannosaurus is a carnivore. It inhabited North America and is 40 foot long by 17 feet tall. The Tyrannosaurus was an omnivorous creature and inhabited the northwestern part of the U.S. and is about 22 feet long and 13 feet tall. The Allosaurus was also carnivorous and inhabited the U.S. and a country in Europe. It is about 38 feet long and 12 feet tall. Some important terms are reproductive maturity, and that is the age at which organisms can begin to produce offspring. Another important term is the medullary bone, which is a bony tissue that forms before ovulation in the marrow cavities of birds as a calcium source for egg shelling. The questions that were tested in the paper were: did dinosaurs grow and mature reproductively like living reptiles scaled to similar size? And can any comparisons be drawn in the relationship between growth and reproduction in dinosaurs to that of reptiles, birds, and mammals? The methods that were used for testing. The researchers analyzed the medullary bone, they used skeletal chronological analysis, they used histological analysis, and they constructed growth curve models. The analyzed data. Using skeletal chronology, the team was able to estimate the age of death and reproductive maturity. They also used growth curve reconstruction, which allowed for life history in each specimen to be recreated. Finally, through the study of the medullary bone, scientists were able to determine the age at which dinosaurs reached reproductive maturity. Skeletal chronology and the medullary bone were each analyzed, and the results are as follows. The Tyrannosaurus reached reproductive maturity at age 18. The Allosaurus reached reproductive maturity at age 10, and the Tyrannosaurus reached reproductive maturity at age 8. The study also suggests that these dinosaurs were prokaryote and had a high adult mortality. In conclusion, the large body size of many dinosaurs precludes the slow growth rates and long generation time suggested by the reptilian growth models. The reproductive strategy of dinosaurs is surprisingly similar to that of living reptiles. It is also similar to larger and medium-sized mammals, but not to that of small mammals and birds. The reproductive strategy of dinosaurs offers insight into their survivorship and degree of neonatal development. Early reproduction would have allowed the largest dinosaurs to have ecologically reasonable generation time. And lastly, the delayed reproductive strategy in all extant birds likely evolved through decoupled changes in growth rate and duration.
The third paper is on the origin of high growth rates in archosaurs and their ancient relatives. Some background information. Bone histology provides vast information about non-avian dinosaurs. In this specific case, growth rates will be looked at. Some important terms. Bone histology, phylogenetic signal, Archosauromorpha, Archosauriforms, Archosauria, and Dinosauria. The question being tested. 3 different hypotheses on growth rates of Archosauriform and Archosauromorph. First, fibrolamellar complex is basal, the lamellar zonal complex is basal, and lastly, the condition varied with each complex evolving more than once. The methods used for testing. The data analyzed was bone histology samples of Triassic and early Jurassic Archosauriforms, including Scaphonyx, Luperasuchus, and Teleocrater. Here we can see the bone histology samples taken from Scaphonyx, Luperasuchus, and Teleocrater. The results. Archosauriforms grew more rapidly during early ontogenetic stages than basal archosauromorph outgroups, and the fibrolamellar complex shows this. Therefore, this hypothesis is favored over the other 2. Concluding thoughts. Fibrolamellar complex implies archosauromorph were capable of rapid growth rate in early stages of life. The growth patterns settled and flattened out later in life, and histological data leads us to believe that high growth rates were a basal characteristic of archosauriforms. The final summary. Through studying specific fossils, researchers are able to gain a better understanding of the growth and reproduction of both dinosaurs, land vertebrates, and trilobites, marine invertebrates. Concluding statements.